Okay, our next speaker is Stephen Lisle from the Department of Biology at Lund University, and his title of his talk is What Causes Evolution, or Ecological Causes of Evolution. Take it away.
Great, thanks. Can you guys hear me and see everything okay? Okay, so first of all, I'd like to thank the ASN. It's a huge honor to receive this award, and I'm super excited to have the opportunity to present my work here for you all today. And so I thought I would start off my talk with a photo from last year's ASN meeting on Asilomar, because that seemed fitting. And so I'm an evolutionary ecologist, and so my work focuses mainly on two fundamental questions. So, there we go. Okay. So first of all, what are the causes of natural selection? That is, what ecological factors generate relationships between traits and fitness that drive adaptive evolution? Can we identify them? And when we do identify them, are they general, or do they just apply in the specific context in which we measure them? And a related question, but at a deeper time scale, are do these microevolutionary processes that we can measure on the time scales of our own lives scale up to influence patterns of evolution in deep time, that is macroevolution? And if micro and macroevolution don't jive, why might that be? And so I think about these questions specifically in the context of sexually reproducing organisms. And sexually reproducing organisms are interesting because we often see striking sex differences in shared traits or sexual dimorphisms. And so when we think about sexual dimorphisms, we sort of typically think about, I think, sexually selected traits like these beetle horns, but their sexual dimorphisms are actually much more pervasive than that. And we see sex differences in a number of traits, including body size and even feeding morphology and habitat use. And I'm particularly interested in these sort of more ecologically relevant sexual dimorphisms. And so when we think, well, how do sexual dimorphisms evolve? We kind of know the answer, and the simple answer is they evolve when selection differs across males and females. That is, the relationship between phenotype and fitness differs for males and females. And so the sort of next immediate question we might have is, well, what causes these sex differences in selection? And typically, when we think about the ultimate causes of sex differences in selection, we often sort of jump to the Darwin-Bateman paradigm. And this is a paradigmatic view of causality of sexual dimorphism that was sort of originally hinted at by Charles Darwin and was elaborated on by Bateman and others. And in this view, all sexual dimorphisms have their ultimate causal root in the gamete differences that define the sexes. So females, by definition, make few large gametes, and males make many small gametes. And the idea here is that these sex differences in initial gamete investment generate sex differences in optimum mating rate and parental care that result in sex-specific selection on a suite of traits and consequently the evolution of sexual dimorphism. And so we know that the Darwin-Bateman paradigm has pretty tremendous explanatory power. But one sort of thing that's interesting to realize that there's actually an alternative sort of causal paradigm for the evolution of sexual dimorphism that has its roots in community ecology. And that's this ecological drivers or ecological character displacement model. And in this model, the idea is that resource competition between the sexes for a shared resource leads to disruptive natural selection and consequently the evolution of ecologically important sexual dimorphism. And so this model is interesting because it really doesn't depend in any fundamental way on gamete differences or anything like that. And I got interested in this as a graduate student in these alternative models for the evolution of sexual dimorphism. And specifically, I got interested in them through my work on eastern newts, Nothoplamus viridescens. And so these newts are a common pond breeding salamander in eastern North America. And so males and females spend a significant fraction of their life feeding and breeding in ponds and lakes. And when we look really closely at them, we find that the sexes have diverged subtly but significantly in a number of ecologically important traits. And in particular, when we look at their feeding structures, we find that males have narrower gapes and longer lower jaws in comparison to females, which tend to have wider gapes and shorter lower jaws. And we see corresponding divergence between the sexes in microhabitat use in their breeding ponds, growth rates in those microhabitats, as well as interactions with other members of their pond communities. And so all of these ecological sex differences in this species led me to wonder, well, could there be some role for this ecological drivers model Could this be a sort of putative case of character displacement between the sexes? And one thing we can realize when we think about this is that we can actually make a prediction from this ecological drivers model. And the prediction is that when we manipulate the strength of resource competition, we should see a corresponding effect on the strength of disruptive selection that acts in a population. And so I tested that prediction in a series of mesocosm experiments. And sure enough, we find that overall there is disruptive selection that aligns in multivariate trait space with morphological sexual dimorphism. So what that means is males and females at the extremes of the phenotypic distribution have a growth rate advantage over individuals at the mean. And we find that when we increase density, this effect essentially doubles. And so we find that competition increases the strength of divergent selection acting on the sexes. And so this, along with other data, is consistent with this idea that competition could be playing a role in the evolution of sexual dimorphism. And so if we go back to this question I posed at the beginning, so we potentially identified a putative cause of sex-specific natural selection. But is this cause general? So is character displacement between the sexes a general feature of the evolution of ecologically important differences between males and females? And this is a question I'm really interested in, and I sort of tried to get an answer in a recent literature review, reviewing cases of ecological sexual dimorphism. But the problem is not a lot of investigators have really gone out and explicitly tested this model of competition driving the evolution of dimorphism. And I think part of the reason that is because the way this model was originally framed and has been framed in theoretical models is sort of as an alternative to this Darwin-Bateman paradigm. And indeed, that's the way I introduced it to you all today. But I think a more realistic way to think about competition is that if competition plays a role, resource competition plays a role at all in the evolution of sexual dimorphism, I think it's often going to be as a sort of interacting cause integrated within the Darwin-Bateman paradigm. That is, resource competition might act and interact with other sources of sex-specific selection. And so with that in mind, I want to shift gears a bit and keep on the topic of identifying ecological drivers of sex-specific natural selection, but shift from a focus on within species interactions to between species interactions. And I'm going to be talking about some more recent work as a postdoc with Dan Bolnick, working on three-spine stickleback. And so three-spine stickleback are an interesting system for a number of well-known reasons. But one reason I was interested in them, and one reason that Dan's group has really gotten interested in them, is that they're host to a number of parasite species. And in particular, at least in Western Canada, the specialist tapeworm Schistocephalus solidus is a key parasite, and infection rates are quite high in wild populations. And so these are tapeworm parasites, and they're trophically transmitted. So host fish acquire an infection by eating an infected copepod, and the tapeworm then grows inside the host fish's body cavity. And they get quite big. So this worm was dissected out of this fish, and a single host fish can host multiple worms, and they're really quite gross. And so, as you might imagine, this is not good for the fish. And so some populations, perhaps unsurprisingly, seem to have evolved a defense, an inducible immune defense against these parasite infections. And that defense comes in the form of inducible peritoneal fibrosis. And so what that looks like is illustrated here, where we have, we're looking inside the body cavity of a dissected fish. And on the left here, we have a normal fish. And on the right, we have a heavily fibrosed individual. And what's happened here is they've essentially induced the formation of connective tissue that gums up all their internal organs. And this fibrosis is effective in essentially encapsulating the tapeworms and preventing their growth. And we see that when we look across infected fish, we find that those that express fibrosis have reduced tapeworm growth rates and induced infection probabilities. However, this inducible immune defense itself carries a cost, at least for females. And when we look across female fish, we find that those that have high fibrosis levels have reduced ovary mass and reduced fecundity. And so one question I was interested in is, are these costs of this inducible immune defense, which we know are high for females, shared across the sexes? So do we also see high costs for males? And we actually thought that there wouldn't be high costs for males. And the reason we thought that is Males don't make eggs. They don't have to fill their body cavity with eggs. And so we just thought they might be less physiologically constrained by this fibrosis connective tissue. Another reason we thought that the costs of immune defense could differ across the sexes is that we see sex differences in parasite infection rates and tapeworm infection rates in wild populations. And in the populations I've studied, we see higher tapeworm infection rates in females than in males. And this is an outcome of sex differences in ecology or sex differences in diet. But we see no sex difference in fibrosis rates. And so males and females, although they differ in their parasite infection rates, if they are infected, they have the same probability of inducing a fibrosis immune response. And so to really get at this, what's going on here, I had to assess what the fitness costs of this immune response were for males. And to do that, I surveyed 2 populations of stickleback on Vancouver Island. And that involves snorkeling around and finding nesting males. So males defend nests, and so you can observe them easily. And so I captured nesting males that had eggs in their nests. And so these are successfully mating males. And I also captured a random sample of other males and females in the same populations at the same time. And so this gave me all the data I needed to perform a phenotypic sexual selection analysis. And when we do that, we find that there is strong directional sexual selection against fibrosis that acts independent of sexual selection on morphology. And so fish that exhibit high fibrosis levels are essentially never successfully defending nests in the wild across 2 populations. This is just data from 1. And so this shows us that costs are not only high for females, but they're also quite high for males. And they manifest via variation in mating success. And so this was a little bit surprising to us, and we're actually still working on data from some experiments, sort of really trying to nail down these costs. But it was a bit surprising to us that the costs were so high for males, and it led us to ask another question, and that is, why haven't the sexes evolved different sensitivity? So a different sensitivity in which they induce this fibrosis immune response. And so the way this works, or the way we think it works here, is fish get infected with a parasite, and then they can induce a fibrosis immune response, and that immune response can also curb the parasite growth rate. However, this fibrosis response carries a fitness cost. And so given that we see substantial sex differences in infection rates, I was curious as to whether or not the costs we see are enough to sort of maintain this inducible immune defense, given that we know it's so costly. And so to do this, I constructed a simple model of inducible immune defense. And in this model, we can treat individual fitness as a function of the costs of this fibrosis phenotype. Fibrosis also has benefits, but those benefits are only expected to be manifest when an individual is infected. So they depend on infection status. And parasite infection also carries a cost. And so we had 2 sort of phenotypes in this model, fibrosis, ZF, and parasite infection, ZI. And these phenotypes are also functions of each other, so they're reciprocal. And so key here is that fibrosis is in part a function of whether or not an individual fish is infected with a parasite, ZI, and also the degree to which they induce fibrosis in response to that infection. And so this immune sensitivity, I'll call it, so this phi parameter here. And so this is a key parameter we're interested in, the degree to which immune sensitivity is under selection based on costs and benefits of the immune response. And so to do that, we can simply plug these expressions for phenotype back into our equation for fitness and differentiate with respect to this immune sensitivity parameter. And when we do that, we find that there actually is a solution for a single optimum, and the location of that optimum depends, unsurprisingly, on the magnitude of these costs and benefits. And so what that looks like is illustrated here. So when the fitness cost of a fibrosis immune response exceed the fitness costs of a parasite infection, we get relatively limited parameter space where we expect an inducible immune response to evolve. Specifically, parasite encounter rates have to be high, and the benefit of the immune response has to be high. So that's pretty intuitive. Don't know what that's about. Okay. However, when the fitness costs of this fibrosis immune response are less than or equal to the fitness costs of parasite infection, we find that the optimum immune response is relatively insensitive to parasite encounter rates. And so we can get a little bit more insight here by actually estimating these parameters as we've represented them here in this model using the data from wild-caught fish. And when we do that, there we go. When we do that, we find that fitness costs of fibrosis do indeed tend to be lower or similar to fitness costs of parasite infection across males and females from the two populations I've surveyed. Unfortunately, I didn't have enough females from Boot Lake to really estimate these parameters. But this is consistent with immune response optima being similar across the sexes, even when they might differ in parasite encounter rates, which we know they must. And so the take home here is that this data shows that an ecological stressor in the form of an immune response to a parasite can have concordant fitness effects on males and females that act to align natural selection across the sexes. And so that's pretty interesting, but we can sort of continue on this line of questioning here. So we've identified a cause of sex-specific natural selection, or a contributor to sex-specific natural selection. And is this general? And so I unfortunately don't have any data on immune response and parasitism from other systems, but we can ask the general question, does an environmental stressor act to align selection across males and females? And this is a question I do have data on from a recent meta-analysis that I was involved with. And this was a collaboration with Deb Godert, Aaron Reedy, and Eric Svensson. And so what we did, what we were interested in is to understand how abiotic environmental stressors might influence the sex specificity of natural selection. And so to do this, we compiled studies where investigators went out and measured natural selection on the same trait in the same population at the same time in both males and females. And then they replicated that through time or space. And so we matched this up with environmental data from the same time and space, spatio-temporally matched. And we had data on temperature variation, precipitation variation, and evaporative potential, this PET variable down here. And so with this data, we're able to perform a meta-analysis to see the degree to which these variables act to align or not align sex-specific selection. And so the results are kind of complex, but one sort of simple way to look at it here is in these effect estimates. Negative effects mean that that environmental variable is reducing the sex difference in selection. So it's acting to align selection across males and females. And the only significant effects we found were acting to align selection. And so and I should say the data here come from a variety of plant and animal taxa. And so this is sort of a look, a test of this general idea of whether or not environmental stressors act to align selection in males and females. And it seems based on this like they might. And so finally, in the last part of my talk, I want to sort of scale up a bit and ask whether or not the microevolutionary processes that I've been talking about might scale up at all to influence patterns of evolution in deeper time. And if not, why? And I really think this question of connecting micro and macroevolution is one of the sort of, I think, most intriguing and sort of biggest unsolved question in contemporary evolutionary biology. And so my interest in this really started with some of my work, again, on salamanders that I'm going to jump back to for a moment here. And if you'll remember, I presented work earlier in my talk showing that there's ecologically important sexual dimorphism in one species, that was actually in one subspecies, Nothopolis viridescens, and that there's evidence from a field experiment showing that there's competition-driven disruptive selection that might be underlying the evolution of this sexual dimorphism. But one interesting sort of point to realize is that that was just one subspecies, Nothopolis viridescens, and it's one subspecies in a clade that I think is a really interesting but perhaps underappreciated example of an ongoing adaptive radiation. So this Nothopolis genus has radiated, there's only 3 species, but 6 lineages that have really sort of exploded since the last glacial maximum. And importantly, all 6 lineages share similar ecology. So they're all pond breeding salamanders. So males and females are spending a significant fraction of their adult life feeding and breeding in ponds. And when we look at the evolution of sexual dimorphism across this genus, which I'm showing you here. So I'm just showing you data on 2 traits. So this is jaw length, and this is snout vent length, the measure of size. And the data points here are species means, and they're for males and females connected by dashed lines. And so when we look at this, we can find that there's been substantial evolution of sexual dimorphism, and we can characterize that evolution by plotting this vector of macroevolutionary divergence between the sexes. So this is a vector describing variation in sexual dimorphism across species. And intriguingly, when we overlay this with our estimate of within population disruptive selection, so this is the direction and trait space within a population of one subspecies that describes the sort of maximum curvature of the fitness surface, we find that there's geometric alignment between these 2 vectors. And so clade-wide multivariate divergence between the sexes can be predicted by our within population estimate of selection. And so that's pretty cool. It suggests our microevolutionary studies might be able to tell us something about macroevolution. And it adds to a small but growing number of studies that have related geometrically selection with among species divergence. And so it leads to this sort of final question. Well, that's neat, but how far can our microevolutionary models take us? And I think this is an area of ongoing work in evolutionary biology. And one sort of key result that I want to highlight that's been inspirational for me is some recent work by Steve Arnold and Joseph Ueda and collaborators. And what these folks have done, I think, is really cool. They've taken a massive database of vertebrate body size evolution. And when we look at this database, we see on the y-axis here, we have change in body size, pairwise change in body size. And then on the x-axis, we have the time interval over which that evolutionary change occurred. And what we find is there's substantial stasis until very deep time. But rather than plotting it, Arnold and Ueda and colleagues went a step further and actually fit quantitative genetic models to this body size data. And when they do that, they find evidence for a model of shifting stabilizing selection. So stabilizing selection towards an optimum that shifts with some probability. And so they can estimate that probability. And when they do that, we find the average wait time for a shift in a lineage's optimum is 25 million years. So what this data is telling us is that on average, lineages are sitting pretty close to their optimum phenotype, and that optimum phenotype stays put for 25 million years. And so this is essentially quantified this old paradox in evolutionary biology, the paradox of macroevolutionary stasis. And so one question I had is, well, what might be missing from these models? Because this is at odds with a lot of our microevolutionary studies that we all perform, where we go out and measure selection and strong selection in wild populations. And so this is a question I was interested in with some collaborators, Dave Punzelin, Niall Rawlinson, and Locke Rowe. And so one sort of key feature of real populations that's missing, we think, is
has been really good.
Is demography. So that is the reduced mean fitness for maladapted lineages. And so as we all know, when a lineage is far from its optimum, mean fitness is reduced, and that lineage faces greater risk of extinction. And so to explore the consequences of demography and maladaptation, we performed simulations of this model of a shifting optimum through deep time. And what we did that was different than some past work is we explicitly accounted for demography and the possibility that maladapted lineages can go extinct. And so I don't have time to present too much of that results, but I can just show you some of it here. We find that perhaps unsurprisingly, the probability of lineage survival decreases dramatically with large shifts in a trait's optimum. And so once we get beyond a few phenotypic standard deviations, the probability that that lineage will actually survive to reach that new optimum plummets. And all of this leads to a substantial increase in the wait time to observability of phenotypic change in trait data. And so we can show that here. So the dashed green line is when we don't allow extinction. But when we allow demographically driven extinction, we find that wait times to observe a certain magnitude of phenotypic evolution go way up. And so what this is suggesting is there might be serious survivorship bias in our phenotypic time series. And that when we see stasis, it's not necessarily implying that optima don't move. more rapidly or more often, but that we can only observe sort of the fortunate lineages that happen to reside on stable optima. And so the take home here is that empirically connecting micro and macroevolution might be difficult or impossible, despite the fact that there's shared concepts, especially from evolutionary quantitative genetics, that bridge these evolutionary scales. And so I talked about a lot of different topics today, but the summary comes down to just a few key points. And so in the first part of my talk, I presented data that general ecological factors can lead to both divergent selection in males and females via resource competition, as well as concordant selection via shared immune responses to a parasite. And finally, evolutionary scales can sometimes be linked empirically via the parameters of microevolutionary change, but the details could really matter. And so with that, I'd like to thank a number of people, but especially my mentors, Locke Rowe, Dan Polnick, and Eric Svensson. And thank you.
Thank you very much for that. We have about 4 minutes for questions here. Yeah, I think there's one that just came in. Here's the question. Because environmental stressors seem to align selection between the sexes, might that reduce the instances in which you would find ecological drivers of character displacement between the sexes? Do you think those two drivers of sex-specific selection or sex-independent selection can coexist? When might you expect one to be more important than the other?
Sure, yeah, that's a good question, because I mean, you could think about competition as an environmental stressor. I have to say that, it's hard to reconcile with the data. I mean, in terms of theory, these models of character displacement are actually, that disruptive selection only happens when a population is on or near its resource optimum. And so if you think about stressing a population in terms of it being maladapted, we actually don't expect that competition-driven disruptive sort of feature to occur. But in terms of the actual data, those are difficult sort of features to disentangle. And so, it's really hard to know with these meta-analyses what's going on. I was actually surprised to find anything in that meta-analysis, because even though that it seems like it has to be true if you pull males and females way away from, even if they have different optima, if you pull them way away, selection has to be aligned. But there's a million reasons you might not see that.
So one of the questions I had, so in the start when you were talking about teasing apart the role of what I think of as traditional sexual selection versus differential ecological competition, and then but the inferences were made on how changes in density affect the effects on males and females. Do you think you can tease those two things apart with that? So like I work on fish, and if I just go and increase the number of fish in a tank, they're going to fight more, right? And then females might get harassed by males more. So do you think you're really getting at ecological competition versus are you altering the rates at which males and females interact as a function of density?
Yeah, that's true. It's really hard to independently separate those. And I mean, in a single experiment, I don't actually know if it's possible. I do have to say that we also find that selection is frequency dependent in the manner we'd expect under resource competition. So I can't basically say that it's only competition there, and I don't think it actually is. I wouldn't deny that there's certainly a role for sexual selection in this system, and also the time scale of those experiments were sort of designed to be slightly off from the mating season too. That a bit.
Yeah.
But yeah, that's a good point. Sort of a big caveat, actually.
Another one that just came in. Have you considered how among species Wrench's rule might be explained for cases like hummingbirds in which sexual size dimorphism covers the entire spectrum from males less than females to males greater than females?
Okay. Yeah, so Wrench's rule is this macroevolutionary pattern of allometry of male and female size, where it basically boils down, so Daphne Fairbairn pointed out that it boils down to higher rates of evolution of male body size than female body size. And so we often see that, especially in clades where males are larger than females, and then in clades where females are larger, we don't always see that. And so, yeah, I have a paper from a while ago where I sort of made the point that it's hard to it's hard to make a prediction with Wrench's rule, but it also falls apart a lot. At least I had data from amphibians showing that it does fall apart in female biased clades. We actually find females are evolving body size at a higher rate. Body size evolution is higher in females than males. And so how you get higher rates of body size evolution in males, even in clades where females are the larger sex, I mean, I think there's probably a lot of different ways that could happen. Yeah, I mean, it's sort of that's like a real challenge. It's just a correlative pattern, and it's hard to explain. So
Thank you very much. I lost track of the time a little bit, so I think we need to stop it there. But yeah, thanks again for a nice talk.
Okay, let's everyone thank Stephen for a great talk.